This video is an overview of the cell cycle. Before we can really get into what the cell cycle is, we have a little bit of terminology that we have to discuss. So we're going to watch a quick video to talk about DNA and how it is packaged within the cell.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
So let's further define the terms that were used in that video. Now, if you have not already printed off the note template, you might want to do so, especially for this lesson, as there are some diagrams on the note template that we will be labeling. If you are very artistic, feel free to draw them or sketch them on your own, but the note template might come in useful for this lesson and the next one. So the first term that we want to take a look at is histone. Those are structural proteins. They're indicated here in blue around which DNA wraps. Now the reason that we have that is DNA is so long, and if we can't wrap it around something, it is likely to get tangled and damaged. Think about it like a spool around which you'd wrap thread. If you didn't have a spool to wrap thread around, you'd have this giant clump of thread that would be knotted and tangled and really hard for your cell to then process or access any of the information that's actually on that DNA. So histones are the proteins around which the DNA wraps itself to keep itself untangled. Now, any section of DNA wrapped around histones is referred to as a nucleosome. And chromatin, so this entire section here, would be referred to as chromatin. That is a large segment or portion of DNA and the structural proteins that it is wrapped around. Okay? Chromatid was another term that was mentioned. That's when chromosome has duplicated. So if we look over here, this entire structure would be a chromosome. This would be a chromatid on the left, and this would also be a chromatid on the right. So before the cell cycle and cell division happens, we have to have a duplication of DNA so that each new cell gets a copy. So these chromatids are identical to each other, but together they are referred to as a chromosome. They are held together by a centromere, which is a part of the chromatids that keeps them together. And that comes into play and is very important when we start talking about mitosis or actual cell division, because if that centromere is not there or does not form properly, those chromatids don't then separate to either sides of the cell, and we can end up with diseases and mutations because of that. And lastly, chromosome is the highly condensed chromatin. So when we have all of this chromatin and it continues to wind and wind and wind and package itself even more, we end up with chromatin. So another diagram to highlight that is if we start off with our DNA, a double helix structure, we wrap it around these histones. We refer to that as a nucleosome. Those then continue to twist and turn and condense more and more and more and more until we actually end up with our chromosome made up of 2 chromatids held together by a centromere. These structures up here, the DNA on its own, we can't see. It's not large enough to see with a microscope. However, when it is condensed as chromatids or chromosomes, we can actually see them. This is what they would actually look like under a microscope. So continuing with some more terminology, chromosomes, some things we need to know about them is this is where your hereditary information is contained. So all of those things that decide how tall you're going to be, what color your eyes are going to be, whether you sneeze when it's bright outside, yes, that is hereditary, are contained within your chromosomes. Those chromosomes are contained within a nucleus, so we're referring to eukaryotic cells here because we have a true nucleus. It is too valuable to just float around the cell, so they have to be in a protected area, which is the nucleus. And in terms of human cells, we have 23 pairs of chromosomes. You would have received one set from your mother, one set from your father. So those pair up. So you have 2 copies of all of that hereditary information, one from a biological mother, one from biological father. So in total, we actually have 46 chromosomes, but 23 pairs. When a cell is going to divide, each daughter cell or new cell will also have 23 pairs of chromosomes. They need all of the genetic information in order to survive and be a viable cell. So we're talking about non-reproductive cells. So in terms of skin cells, you cut yourself and your skin cell divides to make new skin cells to repair the damage. That new skin cell has 23 pairs of chromosomes. It has all 46 chromosomes. It needs all of that information in order to function properly. So if we had an organism, for example, that had 22 pairs of chromosomes, so 22 pairs, its daughter cells would have 22 pairs as well. So no matter how many pairs of chromosomes there are, the daughter cells will have that same number of pairs of chromosomes. Another term that sometimes comes up is a gene. So if we take that chromosome and if we are to kind of expand it back out again into the DNA, a gene is a segment of that DNA that codes for a particular trait. So if you have blue eyes or brown eyes, it is a gene that codes for that information. Okay. So just kind of a recap on previous lessons is when we're looking at cell division, we have to remind ourselves, why are they so small? Well, the reason is they have to be able to get food and particles and materials into the cell, and then they need to be able to get rid of the waste rather effectively. So the larger they are, the less efficient they are at doing that, because diffusion is a pretty slow process. So as they grow larger and larger and larger, that volume gets grows much faster than the surface area. So when cells get too large in order to be efficient, they must divide. So 3 major reasons why cells divide. First and foremost is for growth. So as you're growing from a baby to a toddler to a teenager and eventually to an adult, the number of cells in your body actually increases. So you have to have division of cells in order to have more cells. Those cells don't just stretch out as you grow, you actually make more cells. So you have more muscle cells, more fat cells, more skin cells in order to allow you as an organism to be larger. The second main reason why cells divide is to repair. So to replace dead or damaged or old cells. So when you cut your finger, you don't just have a hole in your finger forever. Those skin cells actually divide in order to replace the ones that were damaged, as is true with other cells in your body. Third major reason why cell divide is reproduction. Now that could be asexual or sexual reproduction depending on the organism. So asexual reproduction is when there is one parent cell, it divides, splits in half, and 2 identical copies. So each generation is identical to the generation before. Bacteria does this. Some plants do this. We as humans do not do asexual reproduction. We actually have sexual reproduction because we have the fusing of 2 gametes. Now a sperm and an egg are each gametes. They each have half the number of chromosomes. They join together, forming a cell with the full 23 pairs of chromosomes, and all the genetic information is then combined, which allows us to be genetically different than the previous generation. So in terms of the cell cycle, you have this diagram if you print off the note template. There are 2 major parts or divisions with 5 phases. So the first main part is interphase. It actually is comprised of 3 individual phases. It accounts for 90% of a cell's life. So 90% of the time, cells are in interphase. So when you look through a microscope, this is the most common phase you will see. Sometimes it is erroneously referred to as the resting phase because it looks like nothing's happening. that's all. It looks like nothing's happening, but actually a lot of things are happening. So this is when a cell, if it's damaged within the cell, it will repair any damages if it can. This is where it obtains energy. This is when that cell actually grows and expands and prepares for division, because it can't just divide and become smaller and smaller and smaller. Otherwise our cells would shrink. So it has to grow before it divides. And this is where DNA duplicates or replicates itself so that each new daughter cell actually has a copy of the working DNA. The second major part are called the MNC phases, or mitosis and cytokinesis phases, and this accounts for 10% of the time. This is the actual cell division portion, so this little slice of our pie down here. This is when the cell actually goes from one becoming two daughter cells or new cells. There is another phase that we'll talk about in a little bit more detail in a future lesson, but it is known as the G0 or G0 phase. Some cells can temporarily kind of pull out of this cell cycle, so they can either temporarily or permanently stop dividing. This G0 phase is a state of dormancy. So nerve cells will sometimes do this. Other cells in your body, if kind of they've stopped doing any of these, will be what's called the G0 or G0 phase. So in terms of interphase, the G1 phase, which is the first gap phase, this is when the cell is growing. So it starts off kind of small. This is when everything grows. So it is making more cytoplasm, it's making more proteins, it's making more organelles. So Golgi, ER, mitochondria, everything, because the new cells need all of those things as well. So G1 is the growth phase. The next phase is the S phase, or the synthesis phase. It is synthesizing the new DNA. So at this point, when it is synthesized, the DNA is referred to as chromatin because it is condensed. The G2 phase is the second gap phase. And what happens here is you have the initiation of chromosome formation. So that chromatin is continuing to condense more and more and more to actually become chromosomes. This is when you have the synthesis or the creation of enzymes that are going to help the process, help speed up chemical reactions, and structural components that are also needed for the actual cell division that will happen later. And centrioles move to opposite ends of the cell. Centrioles are components that are going to be required to actually pull those chromatids apart and allow them to separate into two cells. Lastly, the M and the C phase, we're going to spend a whole lesson on these two, so I'll just briefly refer to them now. The M and C phase, or mitosis and cytokinesis, are referred to as the actual cell division phase, when the cell actually splits into two sections. Okay, so we have the entire cell cycle broken down into interphase, consisting of G1, S, and G2 phases. Then we have the cell division, or M and C phases, where the cell becomes two new daughter cells.